All right, welcome everyone. I think we'll get started now. Um, Welcome to OEHA's workshop on wildfire smoke toxicity and public health. I just want to make sure that you guys can hear me. Yeah? Thumbs up if you can hear me.
We can hear you.
Great, thank you. All right. So, my name is Leona, and my colleagues Priyanka, Tessa, and Amal and I are your hosts for this meeting. First, a little housekeeping. Please make sure your microphones are on mute during the presentations. If you have questions for our speakers, you can put them into the chat and we will get to them after the presentations, or you can also raise your hand to ask a question. And we should have some time at the very end to get to additional questions. So I would like to start by introducing our first speaker, Dr. Luc Montrose. Dr. Montrose, can you please share your slides while I introduce you? Dr. Montrose is an assistant professor in the Department of Environmental and Radiological Health Science at Colorado State University. His research focuses on understanding the influence of wildfire smoke on peripheral body systems, including the brain and reproductive tract. His lab also studies vulnerable populations, such as wildland firefighters and the elderly, and leverages model organisms to better understand mechanisms of toxicity with the goal of protecting public health. Dr. Montrose, the floor is yours, and we see your slides.
Excellent. Thank you so much for that introduction. Let me make sure what's happening here. Thank you so much for the opportunity to speak today. As was mentioned, I'm an environmental toxicologist in the Department of Radiological and Environmental Health Sciences at Boise State, at Colorado State University, previously at Boise State University. I just moved here 2 years ago. This is my lab currently, recent and current. So some recent graduates and a postdoc that just transitioned out of the lab. But I just wanted to kind of show you some of the folks who contributed to all the research that I'm going to mention today. Probably don't need to do this for this audience. You all live in California, but I'm going to start off by setting the stage with explaining why we want to talk about wildfires and wildfire smoke. This wildfires activity has dramatically increased. We've seen essentially a quadrupling of the acres burned over the last 40 years, and if we look at the biggest fire years on record, these are all in recent memory. I just wanted to point out that wildfire is sort of interwoven into my life. This is a picture of a wildfire that came right to my wife and I's doorstep in Boise, Idaho, that burned essentially right to the community boundary. You all have probably experienced wildfire and wildfire smoke yourselves, and you know that where there's fire, there's smoke, and this is truly shifting the balance of where air pollution in our country comes from. This paper, which is now a little bit old, from McClure et al., was recently recapitulated in Nature, but essentially what it shows is that on the East Coast, air quality is getting better due to prudent government regulation. Things that we can control, things like stack exhaust and automobile exhaust, whereas things we can't control, like wildfires, are driving an influx of particulate matter in the West. And some of our Western communities are already seeing this shift to more than 50% of our annual PM coming from wildfires, and that's expected to be reflected across the entire country in the next coming decades. So all of this is to say that studying wildfire smoke and its toxicity is really, really important. The main drivers of this shift over the last several decades has been increases in temperature, decreases in precipitation, and also, our own forest management decisions that are somewhat different across states. But this image just quickly describes how different forest management conditions can have an impact on the way that fires burn There was no treatment on the far right. There was thinning of trees only on the left, and then there was thinning and prescribed fire in the middle. And so just an interesting thing to think about as we move forward, whether or not we're going to let our fires burn or put them out. So when fires do burn, it creates smoke, and we're interested or worried about the smoke because it contains hundreds, if not thousands, of individual chemical constituents. Some of these can be quite nasty, things that you recognize from studies of cigarette smoke, for example, like acrolein or formaldehyde. And then also things like particulate matter. And when we think about particulate matter as it relates to our lungs, we're really interested in those really small fractions like PM 2.5 or 2.5 micrometers in aerodynamic diameter, or smaller because they get deep into the lung. And we're all obligatory breathers, so we all need oxygen, and that gas exchange occurs in the alveolar space, and particulate matter can do damage to that space. When we think about fires burning, a fire isn't a fire isn't a fire. And so smoke coming from these different sources may all have different toxic profiles. So we need to also understand that this is a very complex field to be a toxicologist in. Things like what's burning, what's the temperature, what are the meteorologic conditions, is it smoldering, is it flaming? All of these things can have an impact on the toxic profile. So you'll hear more about this, I think, from my other colleagues, but to set the stage for sort of toxicity in animal and in vitro studies, we need to understand why we're studying this. So what is the human observations that we've made? We see acute exposure concerns like increases in hospital ER visits. Folks who have asthma or COPD are going to have exacerbations. And folks who are exposed to high doses can have reduced lung capacity, and this can last upwards of years, as we've seen in studies like those done by Chris Malaccio in the Sealy Lake area of Montana. Long-term, though, we don't know as much, but we're starting to understand that cardiovascular disease, stroke, and cancer are all potential long-term concerns. I'm also going to try to convince you, at least in my research area, that peripheral organs are also of interest in studying. So all of this we take into consideration, and we look into our community, and we try to understand who's the most vulnerable. The typical groups show up here: young, old, pregnant, comorbidities, but we also don't want to forget about those folks who work outside and can't get away from the smoke. They can't work from home when it's a smoky day. So we want to try to think about them as well when we're trying to understand toxicity. So my lab takes sort of an interesting approach to this. We look at wildfire smoke health effects from cell to society. Across the bottom there, you can see all the different model systems or types of people that we do research on. And of course, I'm not going to be able to talk about all of these today. I'm just going to throw this slide up real quick in case something is of interest to you. Please reach out. Be happy to chat more about this. We study cows as sentinels. We've done a bunch of community infiltration and vulnerability studies. We do science needs assessments for wildland firefighters. Bottom left there, we just constructed Colorado's first whole body smoke exposure system. You have several there in California, one at UC Berkeley. And so the last one that I'll just point out is we're really excited. We just got an R21 to study wildland firefighter reproductive health effects. So I'll leave that there. Please reach out if you have more questions on any of this. I'm also going to give a caveat that I was asked to talk about wildfire smoke toxicology as it relates to ambient air. Like, how does wildfire smoke compare to ambient air? What's the research on rodents and in vitro? And I could spend a day talking about this. So I'll point you toward a recent publication that we put out, a book chapter on toxicology, because I'm not going to be able to hit all the high points today. So to quickly summarize what we know in the literature right now from an animal model perspective, looking at studies that have used wildfire smoke exposure, we see that there's acute lung toxicity, modulated breathing patterns, transient blood pressure spikes. And then if we look chronically, we see both up and down changes in LDH, which is a sort of an output for cytotoxicity, total protein changes, bronchial alveolar lavage fluid cytokine levels seem to go up. And then we also see differences up and down in lung compliance or lung function. And I want to point out that the reason that we see some mixed results here likely has to do with the way that these studies are conducted and sort of the lack of consensus across our field right now. It's sort of the wild, wild west, and it's understandable in some ways because this is a fairly new field. But you'll see a lot of differences in route of exposure. So I'm kind of presenting that here in the pictures. We can take particles off of filters and then put them into the mouse via instillation. We can expose them through whole body chambers. We can expose them through nose only. We can expose them for hours or months. We can expose them to micrograms or even milligrams per meter cubed. Some people burn what would be representative of the tops of trees, so like needles. Some people burn whole wood. And other people use concentrated air pollution systems where they're actually taking a real world fire that's occurring. They're pulling the smoke in from outside, concentrating that and putting that to their mice or their cells. And so you can see that the exposure system conditions, what they're modeling can all be very different. And so it's not unsurprising that we would see this type of variability across study findings. So one of the first studies that I'm going to discuss with you is from Chris Malaccio. It's a little over 10 years old now. This was a really, really interesting study that Chris did. He exposed mice to smoke, and he then pulled macrophages from those mice. And so he did some in vivo, but also ex vivo experiments. And essentially the overall takeaway here is that he showed immune dysregulation. And he did this, his system is, they actually burn in a wood stove. They have an EPA certified wood stove and a non-EPA wood stove. They burn outside and then they bring that smoke inside through a number of different dilution chambers, and then they're able to expose their mice. So he exposed these mice, and then he hit them with sort of a second exposure with bacteria. And he showed that the higher exposed you are to smoke, the more bacterial burden you have in your lung. And this sort of tracks with some of the new studies that have been shown showing that flu rates go up in heavy wildfire smoke years, and also some folks were more likely to be heavily impacted by COVID following wildfire smoke. Now, the reason for that, he then looked at mechanism, and he showed that macrophages that had been exposed to smoke were less capable of functioning as antigen-presenting cells, so being able to ramp up the adaptive immune system that would follow the innate response. And then he also showed that those same macrophages were less able to do their phagocytosis. So that, think of macrophages in the lung as being the sort of trash trucks that go around and pick up trash. Essentially what he's showing here is that if you looked in the lungs of these mice, there'd be a lot of trash sitting around because the trash trucks are all out of order. This is another more recent study conducted at Northeastern University with a different exposure system. They use a quartz tube exposure system here, and then they're able to do a whole body exposure on their mice. I should mention, as far as dosing, the last study was a 2-hour exposure of 10 milligrams per meter cubed. This is a very different paradigm at Northeastern. They're doing 22 milligrams per meter cubed, and then they're doing a fairly long exposure. So 2 hours a day, 5 days a week over 8 consecutive weeks. And part of the reason that they're doing this is they're sort of trying to emulate about a 10 to 15 year firefighting career. So this is supposed to be an occupationally relevant exposure. They're able to show cardiopulmonary dysfunction, including thicker and stiffer aorta and reduced ejection fraction and airway thickening, shown here in the red boxes. So in our lab, we've taken that information that I just sort of gave you a snapshot of, which is that there's immune system dysregulation, there's dysregulation of the cardiopulmonary system. And we're trying to take that sort of a step further. And what we've shown here is that wildfire smoke is also associated with epidemiologic findings like dementia. And some animal studies have shown that neuroinflammation is possible following smoke exposure. And so a few years ago, we put out this model, postulated model on the left, which is a mechanism whereby something you breathe in actually has an impact on your brain. And what we're showing is that there's multiple ways this could happen. The particle could go directly to the brain up the olfactory tract. It could go into the lung and then be liberated from the lung into the circulation and reach the brain. Or maybe the particles don't need to get to the brain at all. They stimulate a systemic inflammatory response, and it's those inflammatory mediators that have an impact. Either way, what we're saying is that somehow the blood-brain barrier integrity is disrupted, and either the particles or the cytokines, the inflammatory mediators, are getting into the brain and causing inflammation. And so this is our sort of postulated idea here for that. And similarly, I mentioned that we can have inflammatory mediators gain access to the circulatory system. If that were the case, then a number of other organs might also be vulnerable. And we're saying that we think the reproductive system may be particularly sensitive. And we're seeing some observational data suggest that this may be the case with a study out of Oregon, which showed that repeat sperm donors were impacted by wildfire smoke. And this was somewhat followed up on by some folks in Russia who showed that peat smoke exposed in male rats specifically, when those male rats were bred to naive females, their offspring had changes in behavior. This would suggest that there's some impact to the sperm, and that sperm is conferring a deficit to the offspring. So in our lab, what we've done with this information is we did a direct exposure of low dose smoke to mixed glia. And at 6 and 12 hours, the data was only starting to get interesting, but at 24 hours, sort of a graph that a toxicologist dreams of, this really beautiful dose response curve here, that as you go up in these half-log dilutions at very small, what I think are biologically relevant doses, we're seeing a decrease in cell viability in these mixed glia cells, these cells that are resident to the brain in this mouse model. We've also taken this information thinking about the reproductive health effects, and we've used a microscopic worm model. We use these worms because they have in vitro-like use of ease. They have a very short lifespan, and we're able to do high throughput tests for dose, duration, source, and mixture. So we really like this model. And it's also interesting to point out that there's a strong concordance between what happens in a C. elegans egg viability assay and what happens in mammalian development in future studies. About 89% of the time, there's concordance there. And so what we've shown is that as you go up in dose, you kill these worms off earlier. We can affect their movement, which is probably a marker of neurotoxicity. It's a dose response curve here that shows the more smoke we add, the more lethargic that these animals get. We show that we can reduce their velocity, which is correlates with health span. We can also reduce their pharyngeal pumping, which is how they breathe and eat. And that's a correlation with neuromuscular function. And we've also shown that if you expose the adults, you let them lay eggs, and then you raise those progeny up, we show a very similar dose response curve over here with these head swings. Like I said, this is a mechanism showing neurotoxicity and lethargy. We're able to see that the progeny of the exposed adults are more lethargic themselves. In our male mice, we're able to show changes in expression patterns in the brain. And these expression patterns have been shown in other people's studies like Matt Campin's group at the University of New Mexico. So we think these expression patterns that are reminiscent or tied back to a lot of neurodegenerative diseases, that these expression patterns are quite robust, and we can have confidence in these because not only did we see these in our study, but we also saw them in a completely different study where the exposure paradigm was pretty different. In those same mice, we also collected their sperm, and we were able to show that there were DNA methylation changes in those sperm patterns. Essentially, the sperm DNA methylation that was supposed to be lowly methylated was shifted upward, and the ones that were highly methylated were shifted down. And these small changes can have huge impacts on expression. Interestingly, though, you might think, well, if it's in the sperm, DNA methylation patterns should get erased when they go to become part of the zygote. We actually found that among a group of gene patterns that are likely to escape that erasure, we actually found that 20 of those sites were differentially methylated, suggesting that they could be passed on to the offspring, and of those, 15 were actually conserved in humans. And one of the last things I'll leave you with that we're trying to think about in our lab is, if you think about how people are exposed to ambient air pollution, that is a very different profile to how people are exposed to wildfire smoke. And we need to start thinking about these different community level effects as well as occupational effects, and how do we model that in animals, and then bringing back those challenges that I mentioned earlier about why our data may be variable across study. How do we better find consensus, or at least be able to talk about these in a way that we can interpret across studies? My selfless plug is, if you're interested in all of this information and how it translates to human health, for the last 4 years, this is the 5th annual Rocky Mountain Wildfire Smoke Symposium, founded in 2020, platform for dissemination. There's also a hybrid option this year. Would love to have folks from California bring their sort of lessons learned and their perspectives to this meeting. This is one way that I'm trying to shift the field forward. So with that, I'll stop there, and I'll be happy to take any questions if there's still time.
Thank you, Dr. Montrose. That was really great. And we do have a couple of minutes. I don't see any questions in the chat, but if anyone wants to raise their hand and ask a question, we can do that. I think I see someone, Anatoly. Would you like to go ahead and unmute and ask your question?
Sure, can you hear me?
Yes, we can hear you.
I can ask a quick question on C. elegans. So AIHA works with Dr. Jennifer Fung of UCSF on developing in vitro high throughput reproductive screens. And her lab worked on C. elegans. And they specifically developed this one assay in C. elegans that's high throughput, based on the observation that reproductive toxicity in C. elegans results in higher ratio of males in progeny. I was wondering if you saw that, or just kind of just an interesting connection, I thought.
Yeah, it was on the slide, but I didn't mention it to the group that these are mostly hermaphrodites. But there's a random selection for about 1 to 2% that are male only. And you're right, I have seen that some environmental exposures can actually shift that percentage of random selection for male only. So that would be a really interesting sort of output to look at. Thank you for that suggestion.
Okay, wonderful. Let's see. Does anybody else have any questions they would like to ask at this time? All right, I don't see any more hands or anything in the chat, so I can pass it back to Leona for our next speaker. Thank you.
Okay, thank you so much, Dr. Montrose, and thank you, Priyanka. So our next speaker is Dr. Lisa Miller. Lisa, or Dr. Miller, while I introduce you, can you please share your slides? Dr. Miller serves as the Associate Director of Research, the Cardiorespiratory Diseases Unit Leader, and Inhalation Exposure Core Leader at the California National Primate Research Center at UC Davis. Dr. Miller's research is focused on investigating the impact of environmental exposures on pulmonary and immune system development during early life. She uses animal and cell culture models to address questions related to mucosal immune mechanisms in pediatric populations, emphasizing an understanding of susceptibility to lung disease in children. Dr. Miller, I can see your slides and the floor is yours.
Thank you so much. Am I coming in clear? Loud and
Yes.
clear? Okay,
Yeah,
awesome. Okay, first of all, thank you so much to you and Priyanka for inviting me to present some of the research that we're doing here at the center. Oops, and here we go. Okay, and thank you to Dr. Montrose for perfectly setting up the presentations, at least my presentation, and I'm sure the follow-up presentation in terms of providing background on why wildfires are a real problem and why we are so concerned about public health. So, and just as a follow-up, starting with the problem, right? In terms of wildfire events, and I think Luke perfectly illustrated the issue here. It's not so much the number of wildfires, but it's really the severity of wildfire events and the fact that over time, the number of acres burned has increased dramatically, and this is what's caused much of the destruction and the contribution of wildfire smoke on human health. So clearly, wildfires impact damage or impart damage to property, as well as can be directly injurious to humans. But the smoke produced from wildfires, as again, Dr. Montrose discussed, contributes to significant health effects. And this is due primarily to the amount of air pollution that's produced from wildfire smoke. And again, I'm preaching to the choir here in terms of, recognizing the amount of air pollution being produced by wildfire smoke of wildfire events. And so the EPA recognizes wildfires as a major source of air pollution. And recent, all of the data we have thus far indicates that much of the air pollution, at least on the western side of the U.S., is contributed by wildfire events. So, I think Luke really touched on this, the challenges that people who investigate wildfire smoke health effects. The real question is wildfire smoke any different from other forms of air pollution? And I think the data at this point in time are sparse, primarily because of the challenges, as again, Dr. Montrose illustrated, of experimentally defining, why or if and why wildfire smoke is more toxic to human health. I'm just showing one piece of data which was published a few years ago from a group down in San Diego, and it's an epidemiologic study that shows that wildfire events that take place in so this is Southern California, and in this case the group tracked not only the location of the wildfires, but the direction of the smoke plumes, and demonstrated very clearly that the smoke plumes were moving based upon weather patterns, moving toward the more central part of California, and correlated that with quite a striking increase in respiratory emissions, and compared that with other forms of air pollution in the area, and basically discerned that wildfire smoke events, the air pollution from wildfire smoke, appear to have a greater impact on respiratory health as measured by admissions and hospitalization. So why is that? And again, I think Dr. Montrose really touched on this. The smoke from wildfires is incredibly toxic complex mixture. And one of the contributing factors to this toxicity and the variability is the fact that wildfires don't just burn biomass. And there's actually a term that describes this combination of biomass and man-made materials, and it's due to this what's referred to as the wildland-urban interface, the transition between wildland and human development. And so what happens during a wildfire event is that both biomass, and the biomass can be a mixture, which again adds to the complexity. The combustion of both biomass and man-made materials creates toxic smoke plumes that again are challenging to characterize. And in addition to creating toxicity at the site of combustion, the plumes can be carried hundreds of miles away. And just to illustrate how devastating and toxic, potentially toxic, these wildfire events are, an image that those of you in California are probably quite familiar with. It's a very tragic wildfire event in Paradise, California in 2018. It's referred to as the Camp Fire. And what you're seeing here is all of these, this area here is consisted at one point of a neighborhood. So the homes were completely destroyed in that event. And so just to illustrate how and again, this is observational evidence, not experimental evidence, but just to illustrate how these differences in structures may contribute to differences in the air pollution and the type of air pollution being produced. I'm borrowing data from the California Air Resources Board. This is actually incredibly informative. What they show here is that, and so they provided data on wildfire events that took place in 2018. So these were all occurring within a relatively similar time frame, all occurring within Northern California. So the type of biomass is most likely very similar. And they compared the number of acres burned, and then look over here on the far right, the number of structures burned. So the Camp Fire by far was the most destructive with regards to structures. Compare that with the Carr Fire, which took place a little bit earlier in the year, much more destructive in terms of acres burned, but not nearly as much destruction with regards to structure. Now compare that with the amount of PM 2.5. So if you just look at all the PM 2.5 as a whole, the Camp Fire over here on your right produced I would say dramatically more PM 2.5 during this relatively short period of 2 weeks relative to the Carr, Mendocino, and Ferguson fires over here earlier in the year. And again, it's correlative, it's not experimental, but the major differences between these two, despite the fact that the Carr and Mendocino and Ferguson fires destroyed significantly more acreage, the major differences between these two is the fact that the Camp Fire destroyed a significant amount of structures during this period of time. So, and I'm not going to go through all the data again from this, from the California Air Resources Board report, but I found it incredibly informative and quite frankly, a little scary if you think about the toxicity of these materials. So this is just a fraction of the data. One of the things that really struck me when I looked at these data was that the PM 2.5, the speciation data from the PM 2.5 suggested that metals were quite high, again, in association with the Camp Fire, not so much with the Carr, Mendocino, and Ferguson fires. So the two that's, again, stuck out at me were lead and zinc, but there were other metals that were elevated in this PM 2.5 mixture. And note also that these were collected not necessarily near the site. Chico is probably the closest site. These are hundreds of miles away from the burn site. So again, humans in these areas are getting exposed to the toxic smoke plumes coming from these burn events. So here's the challenge again, trying to how do we define how wildfire smoke influences human health, both on the short-term perspective as well as the long-term perspective. And so these images, as you can see, show that smoke is not the smoke is not just an issue for individuals who are immediately adjacent to the wildfire area. In fact, the smoke is hundreds, if not thousands of miles away, as illustrated in this image from New York, as well as in Minnesota. So these are events where smoke was coming from Canada. And then this is also happening on a global basis. So how do we study the humans in this area? And how this affects their health. So this is quite challenging for folks who do epidemiology, right? There are ways to actually make measures and make estimates of air pollution exposure. What we've done at our center is actually taken advantage of the fact that we have a large group of animals living outdoors, and fortuitously we have California Air Resources Board monitoring site less than 3 miles away, which we've capitalized on or leveraged to provide estimates of air pollution exposure for animals that live outdoors here at the center. So this is UC Davis, if you're not familiar with, this is the main campus, and then the California National Primate Research Center is located about 5 miles away. So, so what does the center look like? So the California National Primate Research Center, I have to watch the time, it is one of 7 national primate research centers. This national designation means that we are supported by the National Institutes of Health. So the National Institutes of Health, the federal government, provides a significant amount of infrastructure support to maintain our colonies. The California Center, because of the weather that we have and the space that we have, is one of the major breeding colonies for all of the national primate centers. We conduct translational research, which includes infectious disease, behavior, and then respiratory diseases, which is what I study. So these images that you see are of the breeding colony, the outdoor breeding colony. So many of our animals, at any point in time, we have between 4,000 and 5,000 animals on site. So it's a lot of animals. At any point in time, about half of these animals live outdoors, as I indicated, in our breeding colony. So what does that mean? These are half-acre field cages. We have over 20 of them, and the animals live in social groups of about 100 to 150 animals in each field cages. Again, these are family units, and we know every single animal in every single field cage, which is awesome. So in fact, I'll show you the data later. We can actually track when the animals go in and go out. So for example, if an animal has a health issue, we have an electronic medical record system that tells us when the animal is out of the colony and in our on-site hospital. So again, so one of the unique aspects of having a breeding colony is that we have very predictable reproductive events. We know in the fall, the animals are getting pregnant, and in the spring, the animals are giving birth. And so we have anywhere of between 500 and 600 animals that are born during the springtime. I also wanted to mention, and Luke mentioned this interest in looking at susceptible populations. So my focus is on the early life period, but we also have a colony of geriatric animals, and they actually look geriatric. They have gray hair, and these are supported by the National Institutes of Aging at the NIH. So, so how do we study the effects of wildfire smoke, particularly the smoke that travels hundreds of miles, and in this case into Yolo County, which is where UC Davis is located. So what I'm going to talk very briefly about are some of the health effects that we've observed in our outdoor animal colony as a result of being exposed to smoke from a handful of wildfire events that have, again, due to weather conditions, have brought smoke into the area. So I'm going to start with the 2008 Humboldt Trinity County fires, and then end with some of the data we have on the 2018 camp fires. We also have some findings from the 2020 fires, but I'm not, for lack of time, I'm not going to cover that today. Let's see. So the animals that live outdoors being exposed to the wildfire smoke coming into the area really presented itself as a natural experiment for my research program. So I didn't mention that. So I studied air pollution and the effects of air pollution on early childhood development using monkeys as a model. And historically, I've done this experimentally in our inhalation exposure facility. But in fact, the wildfire events presented itself as a natural experiment. So in this case, the outdoor animals are exposed to wildfire smoke, and we've been able to track the animals using non-invasive measures over a short period of time and a long period of time. And as I indicated, these animals have a very predictable breeding season. So they get pregnant in the fall, get birth in the spring. So when these wildfire events occur, and depending on when they occur, it can have a major impact on reproduction as well as postnatal growth. So the first study that I'm going to talk about that I have the most data on is the long-term study in which we evaluated animals progressively across their lifespan that were exposed as infants to a wildfire event that took place in 2008. So I can't believe we've been studying this for as long as we have. So these animals are now 16 years of age. They're almost geriatric. Keep in mind also that these 2008 animals, the animals that were born in 2008, were exposed to a wildfire that was actually considerably less destructive than the 2018 campfire. And then we tracked these animals over time through adolescence and adulthood. So this illustrates the lifespan evaluation of these animals from birth through adolescence through adulthood. And as I mentioned, we use non-invasive measures to evaluate the animals. Using blood collection, non-invasive physiologic measures, as well as high-resolution CT and activity monitoring to get parameters of health. So I'm just going to give you quick highlights of some of the data we've collected in terms of their immune responses. Initially, we found that animals that were exposed to wildfire smoke had an attenuated immune response based upon their blood sampling. So what this data shows is an assay that we use in my lab, which is a measure of immune function. It's a very gross measure of immune function, but it's been incredibly informative. So basically, we take blood from the animals, and then we bring it into the lab, and then we treat the white blood cells in the blood sample with endotoxin, lipopolysaccharide, which is a component of bacteria. So when the cells are treated with this with endotoxin, they produce proteins that are indicative of a robust inflammatory response. So it's a measure of how well you respond to a pathogen. And interestingly enough, the animals, when they become adults, seem to be hyper-inflammatory, both at 8 years of age and 13 years of age. So this phenotype in which the animals were exposed very early on in life goes through a transition, but you can clearly see differences based upon the exposure. So the wildfire exposed animals, when they're adults, become hyperinflammatory, which is, I think, actually consistent with what we see in humans. Now, as I indicated, we use high resolution CT to look at the lung structure, to get estimates of the lung structure. I'm not going to show you all of the data. One of the interesting pieces of data that we found when we measured blood vessel density, this is with the assistance of Fluida, a company that essentially uses AI to evaluate CT images. And what we found is that the wildfire smoke exposed animals had a greater density of blood vessels, particularly in their distal airways. And this was recapitulated in animals that were 13 years of age. So again, there were structural changes associated with this early life wildfire smoke period. Now, the other fun thing that we did, I think it's fun for me because it's very different from what I normally do, is to actually look at behavior of these animals in a social setting, so in a naturalistic setting. And the way we did this is that we created these collars that using a 3D printer that allowed us to install an accelerometer. So this is basically like a little Apple Watch that's placed here very securely and allows the animals to roam free in the colony, while allowing us to get information on their behavior, specifically their activity levels. And just a short snippet of data here, or a little bit of data here, but which I think is really interesting. So surprisingly, we thought that the wildfire exposed animals would in fact have lung function deficits and would have less activity. But what we found, surprisingly, was that it's the reverse. We found more activity. But the reason we found more activity is because the animals slept less. So there's clear evidence of sleep dysregulation in the animals that were exposed to wildfire smoke, which I think is really an intriguing idea that we'd like to pursue in the future. So I'm going to end up with the last data that I'd like to cover is transitioning to evaluation of animal cohorts that were more recently exposed to wildfire smoke. And we really are interested in following up on what happened to the animals that were exposed to the 2018 campfire. So as I mentioned, the campfire occurred in November of 2018, which is when we had a lot of animals that were pregnant. And so what was found when we followed up, when a group of investigators here followed up on those animals that were pregnant, and I want you to focus in on the red here, the animals that were pregnant actually had fewer bursts, but primarily with the animals that were in their late trimester during the exposure period, which was, I think, quite interesting. And in addition, the animals that were born as a result of exposure or following the exposure, so the animals that were born in the subsequent year, actually showed some really striking behavioral deficits. So, and these are parameters that my colleague, John Capitanio, who is who studies behavior in animals, reported that in fact, the animals that were exposed have reduced memory capabilities, as well as deficits in social behavior. Now, this is the last bit of data, and so hopefully I'm on time here. Last bit of data, but I was very excited to show you this data was collected and analyzed by an undergraduate in my lab. I call him, I think he's incredibly talented in terms of data analysis. So he extracted weights longitudinally in the cohort of animals that were born following the 2018 campfire and tracked them over time. So now we are at the 2-year mark. Is that right? Oh, 4-year mark, excuse me. And he separated them out between males and females. And what he found, and I know it's hard to see because there's so many data points here, what he found is that in the females that were exposed both prenatally and postnatally to wildfire smoke, these animals actually had greater weight gains relative to their unexposed counterparts. Conversely, the males were the exact opposite. So this is why you really need to separate out males versus females, because they can respond in completely different ways. But clearly there is an effect of an exposure, of the exposure on their long-term health. So I'll end us with a summary of what we observed here at the California National Primate Research Center over the course of multiple wildfire events. And I'll start with the long, which is what when I describe the health effects that we've tracked over time from following the 2008 wildfires, with these animals as they are actually moving into late adulthood in geriatric periods of their lives, show evidence of immune dysregulation, lung remodeling, lung structural effects, as well as sleep dysregulation. Now, in the most recent cohort, the campfire cohort, it's clear that there were reproductive effects as well as neurological effects, and now perhaps metabolic effects as a result of this exposure, both for the animals that were in utero as well as the youngsters that were out in the field cages during that period of time. So, and you know, this sort of wraps in conjunction with what Dr. Montrose presented. It's very clear that the toxicity of wildfire smoke is going to vary with each wildfire event, which makes it so challenging to study. So this is why we need to keep researching this area to really understand on a fine level what this is doing to human health, both in the immediate term. So I will end there and with my current group. Thank you very much.
Thank you, Dr. Miller. Wonderful presentation as well. And we have just a minute. I see one question popped up in the chat, and this is from Michael saying, thank you for the presentation. Can you please clarify the exposure conditions for the controls?
So we are heavily reliant on having a year where there are no wildfires, at least wildfires that are bringing smoke into the area. So in terms of our location relative to where most of the wildfire events in California that have taken place, we are at least, I want to say 200 miles from where most of these events take place. But we're heavily reliant on wind conditions that bring smoke into the area. So we've had years where, even though there are wildfire events, because of the wind direction, we've been able to avoid getting exposed. And again, we are reliant on the CARB monitoring data to confirm that.
Great, thank you. Okay, so just in the interest of time, we'll go ahead and move on to our final speaker. Liana, I'll pass it back to you.
All right, thank you, Priyanka, and thank you, Dr. Miller. So our final speaker today is Dr. Scott Fendorf. Oh, did I unmute myself? Great, okay. Dr. Fendorf, can you please share your slides while I introduce you? Dr. Fendorf is the Huffington Family Professor of Earth Science and a Senior Fellow in Woods Institute for the Environment at Stanford University. He is the founding chair of the Earth System Science Department and presently the Senior Associate Dean in the Stanford Dorr School of Sustainability, with a background in soil science, biogeochemistry, contaminants in groundwater, and impacts of climate change. His recent research examines the metal content and chemistry of wildfire-derived particles and their potential impact on humans and ecosystems. Dr. Fendorf, the floor is yours.
Thanks, thanks, Leona. Thanks so much for the invitation to come and speak to all of you. And it's just really exciting to be here and also to follow both Luke and Lisa. And I think they set up things that I want to talk about quite well. As I launch into this, I also want to acknowledge the people that have done all the hard work that I'm going to talk about. There's a number of postdocs in my group that have really been leading the charge here. So Alondra Lopez, Alex Honeyman, Claudia Avila, and Ali Namayande are the particular ones that have been doing just a lion's share of this work. So with that, I'll go in and also do the perfunctory beginning of talking just one slide briefly on the importance of thinking about wildfires. So we probably all recognize this, that overall our air quality had been getting better over the last few decades. And there's lots of reasons why that is, but we'll just leave it that it is, and except for the fact that wildfire smoke has been increasing. And there's a really nice paper that came out in PNAS back in 2021 that used great markers to show the influx of wildfires on the PM 2.5 in particular. And that's, of course, why all of us here are worried about the health effects, and particularly those in the Western US and California specifically. So what I want to do today is Luke set me up, I think, pretty nicely by talking about the different aspects when we think of smoke in general. And we have volatile organic compounds like PHs, VOCs, and just various VOCs. And then we have the PM that we all worry about, particularly the PM 2.5. And what I want to do is talk about going beyond PM 2.5 and explain what I mean by that. One is actually the particle size distribution. It's not a magic cutoff. There are a range of sizes below that, and I want to just briefly mention that. And then I want to go into the other aspect, which is the aspects of the chemistry of the particles that we often don't consider. And Lisa set me up for that. But just one slide to show you that if we look at particle size distribution, this is actually from this year. We've been out monitoring various wildfires, including those up in Idaho. So back to Luke's previous location, but throughout California and Oregon. And we see this trend repeatedly, that if we look at cascade filters that give us a particle size distribution, that we actually get the greatest amount of particles at not less than just 2.5, but actually less than 0.5. And so when we start thinking about the size of these particles, I think 2.5 is kind of our beginning. And then we really need to start thinking down into these nanometer, hundreds of nanometer size ranges, and think about where that's getting into our lung tissue or beyond, directly transferring into the blood, but also then what those particles are carrying, which is where I want to go next. So I want to go into the chemistry of the particles. And Lisa set me up a little bit on this when she was talking about the wildlife-urban interface and the fact that you can look and see things like lead and zinc in the particles. There's also a whole range. I want to just make sure there's a whole range of organic compounds that can also be in there that we need to think through. I'm going to focus, though, mostly on the metals. And so while I already pointed out that PM 2.5 is a great starting point for us, that there's a whole particle size distribution below that we need to start thinking more fully about. The other side is that when we think in general, we mostly are blind to the chemistry of our particles that are even in the 2.5 range right now. And I just bring out that, commonly when I'm talking about this, that almost all of us now have on our cell phones a weather app that's going to give us the AQI, but none of us think beyond that in terms of like, is there a difference in chemistry from one day to the next? And so I want to get into that. I think this feeds in nicely into some of the variation that Luke talked about in terms of what they see, but it also can flavor what you might see in the primates between different fires. So I'm going to want to be very specific here that I'm not going to be talking about the wildlife urban interface. I'm going to talk about just the wildfire side itself. So if we're completely outside of dwellings, it doesn't mean that we're devoid of metals. And in fact, we can see a really big range in the metal content, and I'll try to go into that. But you have things in California in particular where you have high concentrations or relatively high concentrations of things like chromium or uranium, almost always manganese and iron, along with arsenic. And those are engendered from the geology, so the rocks that we're seeing that then lead to the soils, and those also then get up into the plants. And when we're looking at the smoke and dust that's coming from wildfire, it's really a composite of what's combusting, but also what you're getting from convection of that very surface veneer of the soil. And all that ends up making up what we think of as the wildfire smoke. And so what I want to point out here is that it varies depending on where the fire is burning, not only because of the vegetation, but because of the geology. So we can go, say, into the coastal range, and we'll see that we have a lot of the coastal range here that's really has geologies that are very high in metal content, so high in things like chromium and nickel, manganese and iron. And then we can go to other parts of the geologies in California and say the Southern Sierra, where you have mostly granitic materials, and those tend to be fairly low in the metals. So chromium, nickel, manganese now would be quite low. The Northern Sierra getting towards the Cascades, then it all comes back up into really high metal content again. One of the elements that we worry about most is chromium, and the reason is that in the rocks and mostly in the soils, you have a benign form. It's chromium-3, nominally useful in human nutrition. But the problem is that it can be transformed, and the good thing is at general atmospheric conditions, so 25 degrees C, let's say, atmospheric CO2 and oxygen contents, we don't see a lot of transformation. So most of that rock-derived chromium stays as chromium-3 rather than going into the hexavalent form, which, if you are old enough and you remember the Aaron Brockovich movie, that's the bad actor that was getting into the groundwater from a very different source than, obviously, than wildfires. The catch on this, though, is that when you heat these rocks or minerals, or if it's the chromium has got into the plants, which it does, and you get above about 200 degrees Celsius, you start seeing that the reaction of chromium-3 with oxygen becomes very facile, and you start getting hexavalent chromium formed. The warmer you go, so the hotter you go, the worse it gets. I mean, the faster that transformation takes place. And so what you end up doing is you're taking a compound that is fairly benign and moving it into one that is really, really toxic, and particularly through inhalation. So we worry about it in groundwater through ingestion, but much bigger problem is through inhalation. So what I want to do next is talk about looking at the legacy of fires and then use that as an indicator of what can be in wildfire smoke. And so we're going to use, though, what's left behind from a series of different wildfires as a toxin source and tracer. All right, so what I want to come in to is, and I'll give you just a quick history on this. In 2019, there was a paper that came out from a group in Australia that showed that if they took synthetic minerals that had chromium in them and heated them up, they started getting this transformation. That alerted us to the fact that we should start really worrying about chromium in landscapes where you have high concentrations of chromium-3, possibly being moved to chromium-6 during a wildfire. So right after that paper happened, we had the Kincaid fire, which was in the North Bay. And when we saw that here, thought that maybe this is the place to go test out this hypothesis that the Australian group had put out. So we pulled out quickly the fire maps, pulled out the geology maps and geologic maps, and overlaid them, and realized, sure enough, that the wildfire was burning across material that was really high in chromium and across areas that were really low in chromium, and they could juxtaposition that to the areas that were unburned that had high chromium and low chromium. So in other words, we had this really great matrix to go out and test whether or not wildfires caused chromium-6 to be produced, and whether it mattered if there was high chromium in the rocks or not. And then we followed that up in the next year. So maybe you know the answer to this, because there was a follow-up study with the big fires that we had in the 2020. So for all of you that can remember that, there was in the Bay Area, we really got crushed that year with the CZU fire in the Coast Range, the Santa Cruz Mountains specifically, and then the LNU complex that happened throughout the North Bay. And so we use those. Turns out that we have this really great natural experiment where we have this, what we might think of as a tapestry or a matrix of different geologies. So we have some rocks that are felsic, which in the geologic term that we can just think of as being fairly low in metals, and then ones that get to mafic, so that's pretty high, and then ultramafic that are really high in the metals that we're worried about. And specifically in this case, chromium. And then within that, we also see different vegetation types. And it turns out that's important, one, as an uptake mechanism for things like chromium and some of these other metals. But the other thing is that it determines in large part the severity of the fire. So since our fires are almost always during very, very dry periods, we don't usually take into account a large variation in moisture content unless we're looking at prescribed burns. So they're almost really low moisture contents. And by virtue of that, it's really the biomass that's burning, and then also to some degree, the chemistry of the materials. So if you think of chaparral, shrublands, very high oil contents, really, really flammable. We think about grasslands, we get quick heating, but it's actually low severity because it's quick burn. And then when we're moving through some of the forested areas, it can vary, but often usually the more intense or more severe fires. So we have this nice tapestry again of different vegetation types in the North Bay that overlie the geologies. So we can go up and use those then to study how burn severity along with the geology influences things like chromium content. So we put those together, and this is a map then that has been done for looking at the Kincaid and the Hennessy fire, so part of the LNU fire was Hennessy, and looking at the burn severity. And so you can see here that we have variations in burn severity. And again, we overlay that with the geology map so that we can start to see then how the combination of severity and geology map into what we might see in terms of the chemistry of the particles that are produced. So we went up to these variations in preserves that had the chemistry that we were looking for and the vegetation that we're looking for. We looked, again, we're doing hindcasts, so we're coming in right after the fires, usually within about a week, to look at the particles that are left and that have been transported, but I'm not going to talk a lot about the transport part. Okay, so this is where we're going to use then again the dependency of this geology and fire intensity to see what is left behind, even in the very small particles. And so with that, first we're just going to give you the overall results for what we see for chromium transformation. So this is showing you then the hexavalent chromium that's produced on different geologies. So the felsic, again, are the low metals, mafic, moderate metal content, the ultramafics are the high concentrations. And we're looking at its function of soil depth. And what I'm not showing here is the temperature variation, but I'll just give you verbally that the temperature mitigates quite quickly as we're going with depth. So by the time we're getting about 6 to 8 centimeters, with depth, we're back to ambient temperatures. And so that's why you see these concentration profiles. It's basically a heating profile for us. And what you see then is that in all cases, you have increased concentrations of chromium, hexavalent chromium, when we've had a wildfire, regardless of if it was a low chromium substrate or a high chromium substrate. But then not surprisingly, the more chromium we had to begin there, the more chromium we get. So if we go to the right-hand plot here and look at these ultramafic soils, we start seeing that we have really pronounced concentrations of hexavalent chromium being produced after the wildfire. So if we look at that then in terms of what we're going to get on an ultramafic soil with different degrees of fire intensity, what we also see is that there's a big variation with fire intensity, that the higher the intensity of the fire, we start seeing big increases, big jumps basically in the chromium content, and then it varies a little bit with the different types of vegetation. And so on the right-hand side, you can see these A3 on through A8. Those are all just different high-intensity fires, but some of them have much greater ash content that I'll explain that leads to the higher chromium-6 concentrations. Okay, so hopefully what I've established here is that there's a lot of chromium that can be produced from the wildfires. It makes a difference, one, on the intensity of fire, but also on the geology. What I wanted to go into next is talk a little bit more about the finer particles, and then I want to go in specifically to the imaging of some of those particles. So we're going to use the silt size and smaller. So instead of going to the PM 2.5, we're just going to go with 50 microns. That's what we think of as most probable for wind erosion, and then I'll go smaller from there. If we look just in that silt and clay size fraction is the way the soil scientists think about this, but you could think about this as also wind dispersible. We end up getting these fairly high concentrations within the small particles, sometimes a little bit more than in the bulk, sometimes a little bit less, but always in at concentrations that worry us. What's interesting is that even in the low severity fires, we see a lot of hexavalent chromium proportionally in the fine fraction. And then in some of the ash materials, we get just huge, like just astronomical concentrations where we're moving into percent levels of hexavalent chromium, which is obviously really hazardous if you happen to be breathing this material. And I should say that this is actually what's sitting on the surface veneer after the fire. So for anybody who's coming in as a cleanup crew, if you're coming back in to reestablish your home ownership, all those particles are laying in the weight. They also have been moved, and some of them have been wind dispersed, and we're just not picking those up. We have, like I said, air monitoring that has picked it up, but I'm going to leave that off for today for the sake of time. All right, so I want to go next and just show you a little bit of imaging of these particles. So we take advantage that Stanford sits next to a Department of Energy facility that was the original Stanford Linear Accelerator. Particle accelerator can be used in kind of a newer model of it to do particle imaging where you can get chemistry. And so that's what I'm showing here. So on the images on both sides, what we're seeing is just different resolutions or magnifications with the ability to image total chromium versus chromium-6. So the chromium-6 is going to be in the green, total chromium will be in the blue. And what you can start seeing here in the particles, if we start looking at the right-hand side, this is about a 40 micron particle, so it's fairly large compared to what we think of really for the PM10 or PM2.5. But the first thing to really point that out is that if you look, it's enriched in green around the perimeter of it. That's exactly what we worry about, is that it means that the outer part of that particle is really what has been exposed to oxygen and heating, and it's all hexavalent chromium. And that is what we would be exposed to. So even though we think of bulk concentrations as often the measure, what we really care about is really the surface concentration. And this is telling us that the particles are basically chromium-6 particles. When we start going into the finer particles, we start seeing the same thing. And so I won't show you a lot of the images, but if you were looking at this little tiny dot here, that's a particle that's less than 2.5 microns, and it should be hopefully just barely can see it. It's green, it's not blue. And that's the smaller the particles we get, the more greenish they're getting. And the right-hand side is just showing some of the chemical imaging of those particles, but mainly just the visual is sufficient for what we're looking for. Okay, I'm gonna actually wrap this up quickly and just say that we can talk more about the particle movement modeling. Lisa mentioned that these particles can move hundreds of miles. Some of our modeling is showing actually thousands of miles, and doesn't move necessarily coincident with the main smoke plume. So those are things we have to start worrying about as we're going to be predict concentration. But my main point for today is that wildfires are increasing, or particulate matter from wildfires is increasing. We know that the greater intensity of these fires is leading to more transformation of the metals. That's a really toxic outcome. Largely, it's one that we haven't been completely in tune with, and one that I hope that we all start really seeing illuminated so that we can put that into greater fidelity to assess our risk. And with that, I'll wrap it up and just say that again, that there was a whole bunch of people that did this work that I got to talk about. So thank you so much. I'm happy to take questions if there's time.
Thank you, Dr. Fendorf. Yes, right on time. We do have a couple of minutes just for any specific questions for Dr. Fendorf. I don't see anything in the chat at this time, but if anyone wants to raise their hand, we can ask those questions at this time.
You sure you didn't get some jam here? It's hidden somewhere.
Sorry, I think someone is unmuted. Oh, Luke, go ahead.
That was a really nice talk. I was wondering about you painted a picture of first responders or even homeowners entering back into a wildfire scene as the wildfire is now passed and the devastation is left behind and someone goes into their house. What are the potential mitigative measures to remove or to lower the risk to those folks? Because it seems like if it's in the air, it likely got everywhere. So how do you protect people against that?
Yeah, it's a super good question. And for the sake of time, I pulled some of this out. We've also done studies to see how long is the high chromium concentration persisting after the fires. And essentially it persists until you get the next large rain. And large is a relative term, but let's say, something just maybe a quarter to half an inch of rain. So it can last actually for quite a while. And in fact, in the LNU fire, there were areas where there was no substantial rain for 11 months. So that toxicity is sitting there, if you will, waiting for 11 months. So what can you do? There's really only two mitigation efforts that we see possible. The first one is limiting your exposure. So if you are going to either don't go out and stir up the residual soil, or wear a substantial respiratory system, at least an N95 mask. And then the other part I didn't go into, but in the wildfires, there's really not much we can do about this. If we start going to creating lower intensity wildfires, we actually have some ability to control it. So how do we do that? Well, prescribed burns are really the large way. If you can decrease the fuel source or the fuel loads, you'll actually then decrease the fire severity, and that helps then decrease the metal transformations that we're seeing. So that doesn't really help you if the wildfires already come through, but in terms of future management, that's something that we see as one of the only options for really limiting our future risk. Other than that, it's limiting exposure one way or another.
Sorry about that. Okay, thank you. I see a couple of more questions that are coming up in the chat, and now we can kind of open it to questions for all three of our speakers. And I'll go ahead and pass it to my colleagues Tessa and Amal to help facilitate the rest of the questions and discussion.
Thank you, Priyanka. So the second question I see in the chat is, how do distant air measures of CR6 compare with regulatory limits? I think that's for you, Dr. Fendorf.
Yeah, it's a really great question. Unfortunately, right now, chromium-6 isn't usually measured in any of the air monitoring systems. Total chromium is, but not chromium-6. And so there really isn't, to my knowledge, any regulation on that. What we've been trying to do over just the last few years is change that by going and doing our own measurements. But in terms of current air measurements, that I'm not aware of chromium-6 being speciated. Or sorry, chromium being speciated. So it's just total chromium. And then by virtue of that, it's not regulated.
Thank you. I see we have another question in the chat from Andrew. He asks, while prescribed burns are lower intensity compared to high intensity wildland fires, what might be the implications for its expanded use in the West, particularly exposure in WI regions?
Yeah, well, one, the great thing about them is it comes back to a point that Lisa made, is that I only was talking about the metals and the variation in metals that are from wildfires outside of the urban interface. So we have to worry about those, but we get a whole slew of new ones like lead in particular, but zinc as well, when we start moving into dwellings that might be burning. And so the great thing about prescribed burn is that, one, hopefully you aren't getting any dwellings, so we can omit any of the anthropogenic metals that might come in. And then two is because we do it under hopefully we do it under high moisture levels, the severity of the fire is much lower, and by virtue of that, our temperature profile is much lower. That then leads to lower metal transformations and much lower toxicity. The other part too is that we have really now great fidelity at predicting what the toxicity of this material will be under different conditions. And so for the prescribed burns, we can look at the substrates and say, like, all right, we're going to burn across this. It's pretty low metal content. Really, there's very little risk. this one's, now we're going to go across an area that has much higher metal content, much higher risk. For here, let's make sure that everybody that's going to be impacted by the smoke is notified of the potential dangers and what duration that's going to be, and that they can then take measures to mitigate their risk, much more so than we get to the wildfire. So anyways, I see prescribed burns as actually just being a kind of a winning situation all the way along. It's mostly just getting people to accept that it could be in their backyards. Right? Nobody likes that.
Thank you so much. We have another question for Dr. Montrose. Can you describe how you set up the smoke exposure system for C. elegans?
Good question. So I conflated two things. I probably should have been more clear on this. So for our C. elegans, we do not do an air-liquid interface like you might see with in vitro systems. What we're currently doing is using our exposure system to generate the smoke. We put it onto a filter. We do a methanol extraction with a nitrogen stream concentration. We reconstitute that in M9 buffer, and that's how we expose the worms. So sorry I didn't make that clear.
And then the next question is from Annika. She says, thank you for the great presentation. Did I understand correctly that the toxic outcomes map includes long-range wind transport? I was surprised to see such a high concentration in the Pacific Northwest and less in the East Coast of the US. Does chromium-6 tend to be shorter range? I was also surprised to see high concentration around Iceland. Could you speak to that hotspot? Is that from regions in Greenland?
Yeah, I think the question is really going to the last map that I had. That was actually just showing the distribution of high chromium-3 geologies across the world, and then thinking about where you're going to get wildfires that could potentially transform those. So that's just basically it's a geologic map. That's what you're seeing there. One of the things I'll just sounds so doom and gloom, which is why I'm stalling here for a moment, but I guess it's the reality. So if we look at the Canadian wildfires from last year, where you had the unusual scene of seeing New York or Washington, D.C. Having high PM 2.5 or low air quality, let's say. Those fires were well over 1,000 miles. Well, those were burning across areas that did have high chromium content. And so if we make projections on that, their exposure to hexavalent chromium was actually quite pronounced. Now, nobody was there measuring, which is unfortunate, but from our now ability to do some forecast modeling, they were getting quite a bit of exposure. And the interesting thing is that the metal particles often move out in front and a little faster than the main smoke plumes. So the big catch on this, and where we're going to have to get much better and really bring air modelers in to do this, is to know when our risk is greatest. And I think for my own personal sake, what has changed for me is knowing that these toxic metals can be in the particulate matter, is that it alters my risk calculation. And so if we all have that information, then I think it can, at a minimum, it can give us information of when we should be staying indoors, or if we're going out, when we want to wear at least an N95 mask. And that's where we're heading. But there's still a little bit more work to go.
Thank you, Scott. Next, we have a question for Dr. Miller. Have you found any difference in cancer burden among your exposed animals?
That's a great question. We've not looked, but we certainly can look into our electronic medical records database for that. So one of the interesting things about having these animals in our colony, you can almost consider them a cohort of humans that live outside 24/7, but they are physiologically and immunologically very similar. Now, one of the interesting things about our animals that you don't see in humans is that rates of cancer tend to be very rare. And I think that speaks to the contribution of both environment as well as diet. So as environment has changed, I would expect that we might start to see some differences, and it might be taking place. We just haven't. It hasn't occurred frequently enough for us to really, for our vet staff to actually notice it. But great question. I think I think we should be looking.
Thank you so much. The next question is for Dr. Fendorf. Was your team able to measure the ratio of chromium-6 versus chromium-3 released?
Yeah. Yes, and it varies some. Well, it varies a lot. In the particles that are derived more, what we think from the soil and the dust coming up from that surface veneer, it can be the ratio can actually be quite low, maybe less than 2% of the chromium at maximum has been transformed from chromium-3 to chromium-6. It's really on the surface of the mineral. And what distorts that is if you think of a particle, if there's a lot of chromium in the interior, we actually don't care about that so much. And that's where the chromium-3 resides, and the outer part is what's transforming. When we look at ash, that's where we see much higher proportions of chromium-6. So that the ash that's coming from chromium that was in the plant seems to be largely transforming. So upwards of 75% of the chromium there is actually in the hexavalent chromium form.
Thanks. There's a related question in the chat from Michael about if you can share about the lifetime of chromium-6 before it reduces back to chromium-3. This can impact its residence time and potential exposure. Perhaps it is already factored into your model predictions. Thanks.
Yeah, hey, that's a super great question. We don't have any predictions when it's dispersed aerially. For when it's on the soil, we know that under dry conditions where it's just sitting at the very, very surface, there isn't any reductant that it really encounters, so it persists. As soon as it rains, it one disperses, but two, it often is migrating down into areas where we have good reductants. So just the plant material, truthfully, is actually a really good reductant. So any residual organic matter in the soils will help move the chromium back into its benign form. So it's on a side note, we first were worried about it being a problem for water quality. We don't see that really being much of an impact at all. It's really the fact that it's dispersing in the air or sitting right at that very surface and being exposed through inhalation that becomes the problem. So it just goes back to, it really takes, for what's residual on the soil, it takes a rain event to get that to be reduced.
Thank you so much. I do see that we're almost at time. So I just want to thank everyone for joining. The speakers have said that they are happy to stay on for a couple more minutes because we do have a few more questions in the chat. But for those of you who do have to jump off, thank you so much for joining our webinar. I will just go to the next question, which is a comment actually from Dr. Montrose to Lisa, that it would be interesting to use cell-free DNA techniques, also called liquid biopsies, in the primates, as this might catch pre-cancer changes. And then just another question for Dr. Miller as well, if you want to address both, which is regarding the primates wearing tracking collars, do they resist wearing the collars?
So first of all, thank you, Luke, for the suggestion. I think that's a great idea. I put it on my to-do list. In terms of the tracking collars, it's actually a great way to monitor the animals non-invasively. We print them to spec, so every animal has its own unique tracking collar. And so typically we bring them in, put the collars on, and then when they go back out in the colony, usually the others in the cage will check out the collar because it's novel, and then they go off and it just becomes a part of their environment. So it's a great way to monitor activity.
Great, thank you. And then it looks like we have another question for Dr. Fendorf. Are you aware of any air dispersion models that can currently account for the relatively rapid travel of metals in wildfire smoke?
They're under development, so the answer right now is no. But give it another 6 months, and I think you will.
Thank you. Our next question is for Dr. Montrose. You mentioned methylation at 20 genes in mice that might be implicated in transgenerational epigenetic inheritance of exposure, 15 of which are conserved in humans. Of those 15, were there any genes of interest related to immune, lung, or neurofunction? Although this is a small gene set, so may not be able to infer.
Yeah, and again, another point that I probably glossed over too quickly. So these are actually imprinted genes, and that's one of the reasons why they are thought to escape the demethylation erasure. And so to give a little context, the reason why we have this demethylation is, or not demethylation, but reprogramming is every cell serves a purpose based on its DNA methylation profile or its epigenetic profile. And so that cell, in this case, a sperm cell, looks the way it does and acts the way it does because of its DNA methylation profile. Now it's met up with an egg, an oocyte. It's going to become a zygote, and that zygote then needs to become pluripotent and become the billions of cells that are going to be in your body. So it can't go on remembering what it was like to be a sperm. It has to forget all of that. And so that's what the erasure is all about. However, there are genes that are parent of origin imprinted genes. So essentially you have some memory of your parents and grandparents and great-grandparents that are being passed along because you're getting the same allele expressed every time in a parent of origin fashion, either from the male or female dominated side. And so it's these genes that we looked at to see there's about 70 or 80 of them that are known. And of those, 20 of them were hits in our differentially expressed, differentially methylated gene set. And then of those, 15 were conserved. This is a long-winded answer. I probably should have just started off with, I don't know the answer to your question, but I wanted to give some context because I didn't say that they were imprinted genes, and that may help you understand maybe where these genes are situated. Oftentimes, they're involved in sort of lots of different outcomes or pathways. So I can definitely look into that, though. Thank you so much for the question.
All right, everyone. I think that this brings us to the second ending to our webinar. I want to thank everyone for attending, and those of you who stayed to the end and asked the questions, thank you so much. Thank you, Dr. Montrose. Thank you, Dr. Miller. Thank you, Dr. Fendorf. Yeah, good job, everyone, and thank you so much. Hopefully there'll be another. We'll see you soon.
Thank
And
you,
I would like
everyone.
to thank you, Mark and Rima, also for all your support on putting this together.
There's a lot of great comments in the chat, just thanking all of our speakers. So thank you guys again for presenting and having a great discussion.
Very informative, very helpful. Yeah.
Thank you for having us.
I have a few studies I need to go look up now, so I appreciate that too.
Thank you.